So briefly, my lab works on a diversity of projects. Some of them, I'm talking today about the zebrafish model stuff, a lot of behavior and developmental work. But it's a diversity of backgrounds. So some of the students and postdocs in my lab would be from statistics, bioinformatics, toxicology, chemistry. So I like meetings like this with a diverse background because I think it actually reflects what I do, and I think this is a good home for my research, so I'm excited to talk to people here. And thank you to the organizers, the Oki and Rick Paulus for the session chairing especially. I did want to start out with the acknowledgement slide because I am a dry lab scientist, meaning that I don't touch chemicals or wear lab gloves or look at animals or any kind of stuff. So everything I do is collaborative. And today, in particular, this work is with Oregon State University with Robert Tanguay's lab and Lisa Trong, who's his deputy director, effectively the operations manager of that lab. And we work together to bring this data with Skylar Marvel, who's a programmer in my lab, Guozu Zhang, who graduated last year and is now at Social and Scientific Systems, and Michelle Ballack-Meisner, who is actually giving a talk at NIEHS on Friday. So I'm not going to present some of her newer stuff today. I'm doing a little bit of the stuff that we've already put out in a pub that's in review right now. But if you want to hear more from her, she'll be talking on Friday. And she's responsible for all of this analysis that I'm going to show today. So the big deal question that I wanted to ask is, if you see variability in a phenotype or in an exposure following some environmental chemical, or could be pharmaceutical chemical, is that variability genetic, right? So variability, differential susceptibility could be due to a lot of reasons, right? There's experimentation, there's all kinds of things that could happen. But can we chase down when there is genetics that matter? Are those coming to the fore? Can we find some way to show that those are real? And so I'm going to take you through this system that we use, the zebrafish system, where what we're trying to do is, if I think of this most simple equation, which is the phenotype is a function of gene by environment. If I concentrate on each of those three aspects, right, can I really narrow down that phenotype to get a good idea of what am I actually seeing in these results? The environment, what am I actually doing? What am I giving to these animals? How are they growing up? And the genetics is the last part, which is, in one sense, the easiest part because of all the work that people have been doing on this genetic technology, right? I can measure genetics in lots of ways. I can do constitutive DNA variation. I can do RNA-seq. I can do targeted seq. All kinds of options there, right? But so we have to get a handle on all three of these to really understand this question. And then my conclusion is going to be, right, can we actually use this HTS zebrafish data to find evidence for G by E? Because I don't want to introduce a slow process into this, right? I want to do something that's fast and it's rapid that can keep up with these new chemicals coming onto the market and all the chemoinformatic models and things like that too, right? So we want it to be rapid. And then in the end, does it matter for safety assessment? Like why, right? So when I talk about chemicals, the risk posed by chemicals actually differing across the population, there's a few things, this Deepwater Horizon spill, so this was like the Elk River thing that Scott was talking about. In 2000, I think it was 11, if I remember right, before the movie, there was this, natural experiment that happened in the Gulf. There was a human and an environmental disaster, but all of a sudden you had this experiment where a ton of oil was dumped into a very concentrated place. And we made the experiment better by flying planes over it and dropping dispersant on it too, right? And then after that happened, somebody said, is that bad for you, right? So they're dropping tons of this and that's if it's bad. And this is when I was at NCCT, so the team of us, Richard Judson and Matt Martin and all kinds of others stayed up really late for a month and we worked to do this really rapid test of, well, which is worse, the dispersion of the oil, right? Essentially is the question we're tasked with. And then other examples of, if I think about when that Zika thing was happening in Florida, this is actually a quote up here, unfortunately. And the Florida Department of Agriculture said, if you're worried, stay inside when we're flying planes over and dropping mallet on you, right? If you're worried about it. If you're not worried, go ahead, right? So that is a weird risk assessment set of guidance, right? And it's not, and I'm doing it, and I hope there's nobody from the department in this room because I'm not actually making fun of them, kind of. But it's that without information, what do you do, right? So what's worse? Do you want Inca spreading? Do you want NALA drop down? Like, which is worse? Right? You have to make a choice. You have to make a decision, right? So let's give them the information to make those decisions. Let's us, here in the room, give them the power to do that in a more informed way. This is my hometown, Flint, Michigan. This is where I was born, and they have water there, right? And I think we all know about that confluence of things, right? In the risk assessment lecture that I give, actually, that Rebecca Fry organizes at UNC, this is part of what we talk about: is how does the confluence of the regulatory, the science, the public health, and people voting for Republicans come together to make it that a bunch of people got really, really sick, right? How does all those things kind of come together? And we talk about all four aspects, right? Because they're all important, right? So there's no one evil aspect of these. They all work together to change policy that actually affected people's health, right? And the last one is really up here. If this is a new hotel that's our showpiece hotel in right next to NC State University, and I had some collaborators come from overseas and stay the night, and we were going to have meetings the next day, and we got a call in the middle of the night that one of these, it was a man and a woman, she got really sick, right? So she woke up, they had to go to the hospital, we had to cancel meetings, and they were staying in the same, not the same room, adjacent room, same hotel, same age, brand new, right? She got really sick, he didn't. And we're trying to figure out, I thought, well, maybe she's allergic to the sheets, or maybe she ate something weird. And it turns out that what they did is they moved to another hotel where you could open the windows, right? And she was fine, an older hotel. So what happened is all the furniture in this room had that nice new phthalate smell, like the new car smell, right? And things hadn't outgassed enough. And she was really susceptible to this, and he was fine, right? Same age from the same place, right? She happened to be of Chinese heritage. He was of English heritage. So there's genetic differences between them, but they responded very, very differently, right? So that's a real world example of how these things can manifest themselves. So if we know that these kinds of things happen, want to know what can we actually do, right? What are the implications of this for the population? How it prevented that hotel injury, from happening, right? Because it was approved, right? It was above whatever safety or below whatever safety levels was needed, but it wasn't protective for everybody in the population. So one way to do this is this uncertainty factor idea. These are things we build in, and we're supposed to account, for genetics in these, but a lot of times it's uninformed, and it's because the data don't exist, not because people aren't using it, right? But we need to find where those data can come from to do this. And so I have some quotes from various regulatory agencies and papers about what we'd like to actually do with these, and maybe some idea of a chemical-specific adjustment factor where maybe different chemicals have different effects on things, right? So that's one kind of way to do it. But, again, the big question is, can we quantify or identify the role of genetics in individual variability, right? So if we're seeing differential response, can we find some way to find genetics? And I think that an idea is that people probably agree with, but the question then is how, right? So of the systems that, I personally touch, we have human epidemiological data, mammalian data, fly data, piles of cells, and biochemicals, right? So at the extreme end, you're really fast, you're efficient, no one cares what you do to biochemicals or cells, but biochemicals in general, there's no ethical issues. Cells, many fewer, right, as you go up, more issues. But there's also this trade-off for speed too, right? So, and with the way things are going now, this can keep up with high-throughput screening over here, right? We can do zebrafish as fast as we can some of these high-throughput screening systems, depending on the assay. Same thing for these flies that are growing up as well too, right? So it depends on the endpoint you ask, but we can start to keep pace with these. And I would say that if you're doing something like behavior or abnormal development, an in vitro system is a very difficult one to replicate something in a whole development organism system model, right? So that's a difficult challenge, but I think this is one of the additional models that I think is going to be useful for addressing these kinds of questions. And what's good about it is if we're doing something, and I'm going to show you the period this assay happens in, early in development. So if ontogeny recapitulates phylogeny, right? So way down here, everything starts out the same. These are vertebrates growing up, right? And there's this period where there's kind of a constrained patterning and development, and they've actually done some cool studies with gene expression data to show how conserved that expression is during that period, right? So if I'm studying genetics and I'm trying to translate across models, this is a really nice period to look at the genetics and how it goes, right? After that, I come up with different body parts than a zebrafish or a fly comes up with. But in this very early period, not a fly, some other vertebrate, right? A turtle, right? There's this nicely conserved period here where we all go through the same thing, right? And that's where I'm going to ask my genetic questions. So back to the Tanguay lab at Oregon State. So Robert and Lisa did this experiment a couple years ago, I guess three or 4 years ago now, where most of this data was collected with 1,000 compounds, right, with the whole ToxCast phase 1 and 2 set. And this is growing, right? They're doing more. But this system is what I wanted to glom onto, right, to ask this genetic question. So for this system, High-throughput, it takes 5 days, right? So it takes 5 days to run through this. And each chemical is tested in 6 concentrations. You have 32 replicates per dose, right? So what's important there is that they're not pooled, right? So there are some times when pooling is efficient and it makes sense to do, but if I want to do a genetic experiments about individual differences in responses, I really need them to be individuals, right? So they're in individual wells. So each well is its own little microenvironment for those 5 days to grow up in. And we have time series data. They score a bunch of morphological endpoints. Some of these we're helping them automate some of these. Some of them they've dropped, added, and multiple behavioral endpoints as well. So we have a lot of data coming out of this. And if I look at this phylotypic period, right, I'm turning it outside, this constrained period here is actually generally agreed to be somewhere in that 10 to 48 hours, something like that, right? You can move it a little fore or aft of that particular time point, but it's somewhere in there, right? So we're hopefully capturing that period where everything is most similar, right, and coming up and doing that. And so now if I want to do genetics onto this, I can use the same system to ask genetic questions, right? So I'm not coming up with a new assay that we have to modify things. I can just say, while you're doing this, I'd like some DNA at the end, right? So we can keep it going fast, right? The validation work is a little bit different, but I'll talk about that. So what we did, these Zhang papers here. So first, Lisa published the whole data set in a ToxSci paper in 2014. I worked with them. We did some of the behavioral data, some predictive applications there. And then these Zhang ones, this is all GLOSIT, right? So these are methods, analytical methods to analyze these data. So I have this dose response data. I have an early behavioral endpoint. I have later behavioral endpoints. And I have developmental assessment, right? And I'm not going to go into details of this. I can talk later about it because I want to get to the genetics. But the takeaway here is that I have all these data all the time, right? So this course of data gets collected on everything. And then we have analytics that are looking at how's the development related to the behavior, what changes are we seeing, what was the environment, what was their microenvironment. So we're doing a very, very detailed look at trying to capture everything that we can know about this 5-day experiment. What is everything that happens to these animals over that time? Trying to tamp down that epsilon, that error term, in a y equals g by e equation. So now finally, I'll get back to what the genetics is, right? So in the genetics, I'm talking about differential susceptibility. So the examples I gave at the beginning are, a storytelling way to do it. But what the data actually look like would be something like this, where this gray is a response that I would expect in a population. Some sort of normal response for a quantitative trait. In this case, it's a behavioral assay. Some things move a lot, some things move not very much, just normally, no chemicals. Now, if I drop a chemical on it, right, some sort of environmental contaminant or pharmaceutical or something, and it's a bad one, right? It reduces movement. This is, if I'm doing a risk assessment, I say, don't give any more than this, right? This is where it's bad, right? Don't give any more than that because there'll be trouble. But some of them really don't care, right? Some of them tolerate it. They're very, very resilient, right? If I gave a beneficial thing, some things would be good and others wouldn't respond to it, right? That would be a drug response. Here we're toxicology, so it's flipped. But there's this big fat tail out here, a very skewed distribution. Those are the interesting individuals, right? So something that is bad for most of the population, some of the population doesn't care about, right? That's what we're interested in. And it turns out that zebrafish are good to study this because most of the populations are actually genetically diverse, right? So experimentally, that's frustrating versus an inbred rodent model where I know exactly what the genetics are going on. That introduces its own problems. But for this question, it's actually a benefit, right? Because this is what human populations look like, right? It's an unknown level of variability, but it is genetically variable. No two individuals are exactly the same, right? So we're going to capitalize on this to actually ask these genetic questions. And the way we're going to do that is by, more than 10.
I'm sorry.
Okay, all right, so I'll talk a little bit about this then. I have some time. I'm back to my equation here where I have y, g, and e, right? So simplifying the problem, my response, in this case a differential response, is some function of a gene-environment interaction, right? That's the model that I'm looking for. And as in all experiments, there's an error term, right? There's an error term associated with all of these things. But essentially, we're making a big one conceptually, is what are all the other things I can get rid of so that if I see gene-environment interactions, they come to the fore, right? I can actually observe them and not be covered up by this other variance. So what we're trying to do is reduce that by looking hard again at these phenotypes, right? So we have these analytics where we can say, for this animal, it had this particular thing, this other one had that same particular thing plus two other things. Those aren't the same, right? That's not the same phenotype. So one developmental defect is different than three other developmental defects, right? There's correlations and relationships, but we want to understand for each individual exactly what that phenotype is, behaviorally and morphologically. And then we can go to the environment and say, this is actually a real plot of the behavioral assay over a period of a year or so. At different times when they did an assay for a particular compound set. And you can see that they're lining up in a nice, normal way that it looks like even if they do the experiment, we do a retesting round a year later, we're getting a similar response that fits into the other ones, right? This is really good. If I were finding some sort of evidence of not that, it could be something like genetic drift. The colony could have been contaminated by something, right? Any number of things could have happened. But I'm looking at this to make sure that it's the same, right? So we can do this for any data set. I picked some arbitrary behavioral data here to show it. But you can do that. So I'm controlling the environment. So I really know what's happening here. I understand that some of the errors that are happening just because you have robots in a lab moving quickly through data. And then we have this, we made an interface for them so that we can make decisions in real time. So when they do the early assessment, they can analyze the data immediately, make decisions about individuals to take forward into adult experiments, back and forth, right? So rather than emailing us a file and waiting for however many months it takes me and my lab to give it back to them, they can do it themselves, right? So we're developing these analytics in tandem with them. So have control of the environment, have control of phenotypes. Now all we need is this big G piece, right? So for genetics, again, there's a bunch of ways to measure this, but now if I have control of those other two, and I'm trying to reduce all the error about those, I can now start examining that genetics, right? See if I can make that come to the fore. So for here, our approach is, if I distill it into 4 steps, the first and most important one is, rather than saying my favorite chemical is, bisphenol A or some other important chemical that people want to study, what we did instead was look at the patterns from the high throughput data and say, hey, look, are there some where there's a variable response, right? So I see a bunch of them where there's a clear toxicity departure point, where all the animals get sick, and before that they're okay. And then we see some, like this, where that slope is really there, where an appreciable percentage start getting sick at different doses, but at 2 orders or 4 orders of magnitude difference, some animals tolerate it fine and some animals are really sick in the same environment. Right? So that's where we're looking for that variability. That's that differential susceptibility that we're talking about. I'm going to ask genetic questions of that chemical. Okay, so that's the most important part. And we can do this with, as the data grow, right, we can keep asking this question and come up with new ones. You could also pick your favorite chemical, but that's not what we did. And then here, this is a rangefinder experiment. The point of these is that each row is an individual and each block here is a particular dose. So we did, what, 8 doses or 9 doses here at first to try to identify the concentration where the red is kind of half of the picture. And then we took this concentration where it's not quite toxic to a lot of them, going bad for all of them here. In between that, another rangefinder, expanded doses, right? And we're talking about like a 1/10 of a micromolar difference here, right? Really, really small differences. We're trying to be extremely exact to find the ideal, if we can, 50% point. Because what I want is for maximal genetic power, I don't know anything about the genetic structure, so I'm going to pick 50-50, right? That's a default assumption, but maximally powerful statistically if I know nothing about the genetics. So we found the 50-50 point. Then we exposed at that 50-50 point concentration individuals to this same environment. So exposed a bunch of individuals on a plate, included some control individuals on this plate, ran 8 or 10 plates of this, right? So around 600 individuals, 600 individual wells. And we looked at the pattern of response of those, right? So amongst those that were identically exposed, some of them were affected, some of them were not. Right, and that's exactly what we were looking for. And some of them had, there's a few that are going to have an intermediate phenotype, right? So it's not perfect, right? But we're going to take the ones that have the exact phenotypes that we're looking for, this big multivariate phenotype that I'll go over on the next one. And we're going to take those and call those affecteds, right? So this is 1, 2, 3, 4, 5, 6 individual developmental endpoints in my affected, and the unaffected are all clean, right? So again, same environment, some of them really sick, some of them not. And that multivariate endpoint is yolk sac edema, pericardial edema, and malformations of axis, eye, snout, and jaw malformation, right? So those things are coming together to make this phenotype. And what we find is when we look at those and do whole genome sequencing of both sets of individuals, we actually come out with a candidate gene. This is a Manhattan plot. For those who are not familiar with this, where I line up all the hits by chromosomal position, and I say which one had the highest statistical significance associated with this 2-class problem, right? Differentiating unaffected from affected, I get this one gene that sticks out, right? This SNP in this one gene called SOC7 that sticks out, and it turns out that actually makes a lot of sense developmentally, right? So we're happy that it was a named gene, right? And these are small, right? So I didn't cheat here. This is actually the highest hit, right? They're not a bunch of hits, and then we were like, well, it'd be great if SOC7 were in the one. So this is actually the highest hit, right? So the highest one up there, and this is the stupid Bonferroni line of correction, right? Where everyone, the most beat upon statistic ever, where everyone hates it. We could do FDR and find other things, but we're not, because we found one that was really, really high without having to do that, right? So we have other candidates we could test. But so we took that and started testing it and found that indeed there's a proportional difference in this one SNP in the affected and unaffected individuals. And if we take that now to validation, right, and say, well, is there some sort of other biological evidence or signal other than our statistics that do that? An independent replication set, more animals did the experiment again, we get this result again, and then we do gene expression on that. We find that the affected individuals actually have a change in expression for this, right? So this is different than saying that everything having to do with this phenotype is because this one gene expression thing is down, right? So think of all the things that went into this. It's 5 days exposure. We measured gene expression at 5 days. The exposure was early, right? There's all this other validation work yet to be done, right? This is not a complete story, but it is saying that the difference between these two individuals has a genetic signal, and that genetic signal is something real, structural, observable that I can find, and that signal translates into some sort of mechanistic thing, right? So we have multiple lines of evidence saying that, okay, this differential susceptibility we observed, I think, is actually due to genetics in this case, right? So this one, I think, is, I'm convinced by this. We have more actually on the way. Those data won't be ready for a little while. We're actually doing forward genotyping to say can we breed these and do something special with it. But for right now, I think that we have evidence that this susceptibility can be tied to population genetic variation. It doesn't guarantee that every time we see differences it's going to be genetic, but in this case it was, right? But using a high throughput system, and now that I have genetic background data, I can make more informed choices for future experiments. We can start asking questions of other chemicals and very quickly find out, well, is there a genetic basis for this for this difference, right? Can we tie this back to genetics? Using, again, this high-throughput zebrafish system, we can ask these G by E questions, and we can use it to identify compounds with support for differential susceptibility prior to undertaking. Sequencing is the only expensive part of this, other than if you've set up the Tangway lab facility already, right? If that's already a sunk cost and that's done and he's your friend, that's free. But the other part, right, the sequencing part is right what I have to pay for, right? So I understand the privileged position I'm in here to work with that group, but you don't want to spend money chasing G by E if there's no evidence that it's going to be there, right? So let's find the chemicals where there might be some effect. A lot of chemicals are so bad that who cares if the genetic effect is a little bit different because they're terrible, right? But there's these other things that are going to be in between and that we need to be protective of. So if you control that variability, you can find it. And so I think, for the next steps, I talked about the additional validation work. We also have behavioral ones. So this is a CDF of a behavioral assay where by genotype you get differences in behavior, right? So this is a quantitative trait that I can link now to genetics in this behavioral system, right? And it's still the same time scale, still 5 days, right? Still really, really fast. We can start doing nanomaterials. So my student Kim To is here today. This is her project. She has made these response surfaces where the response to a nanomaterial is actually dependent on multiple factors, right? It's not a simple dose response. There's a lot of things that go into it. Now genetics might modify that further, right? So it's a difficult statistical problem, but I think a fun one and an important one if these things are going to be out in the environment. And then just last, this is a little bit of a plug, but if we start integrating these things with other sources of knowledge, right, which is the theme of this meeting, I think we can start making reasonable decisions on how to communicate what this data actually means. And this is for people familiar with, if some of you use ToxPy, we actually, Skylar Marvel wrote a new software suite for this that we distributed now for some beta testing, and the paper's coming out soon. But this one is a lot better, right? It does more, it does fun clustering and stuff like that. So we can start to integrate these things. And that's all I have.
Okay. Thank you, Dave.